So the last structure we're going to talk about is the leaf, and we're going to talk about the tissue arrangement in the leaf and growth and development. And I'm just standing back here so you can stop the video and take notes on the different aspects of the leaf structure. So let's talk about the monocots. The monocots have parallel venation, which means that the veins run parallel to either the lateral length of the leaf blade, or they can go from the midrib outward, sort of like the banana plant that I showed you at the end of the secondary growth video. And if you were to look at a cross section of the leaf head on, in other words, like if I cut it across here, what I would see is the vessels are looking like they're coming straight out at you, and instead of coming in sort of on a sideways angle or something like that. Usually there is one midrib, that is one vein that is much larger than the others, and other than that, they all look kind of the same in terms of size. If you are looking at any of leaf, that is even the eudicot, which is over here, we'll talk about in a sec, the xylem is always on the top. That's because remember that the xylem is in the middle of the vascular, or towards the middle of the vascular vessels, and as the traces go outward into the petiole of the leaf, that would mean that they would end up on the top. So if they start off in the middle, they're gonna go out and be on the top, and then the phloem is on the bottom. One thing special or different about the monocot arrangement is that the bundle sheath cells, which are around the xylem and phloem, that will be the same in eudicots, but then around that is another layer of what they call mesophyll cells. And mesophyll just means middle leaf. There are other mesophyll cells sort of in between that are slightly more loosely packed, but the mesophyll cells make a ring around the bundle sheath cells and then the xylem and the phloem. And you're going to see when we talk about photosynthesis that that's sort of a special feature. Let's see what else about this. The upper epiderm and lower epiderm have, of course, these are epiderm cells. I just drew it as a line. Same thing here. There are cells in there. I just didn't want to make the diagram look too confusing. And of course, in the monocot, the stomata are on both surfaces, or usually on both surfaces, with the guard cells guarding the stomata. Now looking at the eudicot have netted veins. That is, the veins come off of the midrib and then branch out in many different directions, and it has a net looking effect. If you did a cross section across there, then what would happen is in cross section you could see the veins that are coming out sort of in different angles. And unlike the monocot, it has a bundle sheath around the xylem and phloem, and again the xylem is on the top and the phloem is on the bottom, but it does not have another layer of mesophyll cells surrounding the bundle sheath cells. Instead, the palisade parenchyma are on the top here. These are the primary photosynthetic cells because they are full of chloroplasts, and they're very large cells, and they, as they do cyclosis, of course, every chloroplast will have exposure to the sun, but they are very big and in a kind of a tight line across the top there. And then the other part of the mesophyll is the spongy parenchyma. These are kind of airier or more loosely packed cells. And what that means is that the gases can go in and out of the stomata, which are generally speaking on the bottom. Not always on the bottom, but I just drew it that way in this particular diagram. There is also something that in both monocot and eudicot that is important, and that is something we call a substomatal chamber. What that means is there's a little space in the spongy parenchyma right behind the guard cells of the stomata, allowing the more exchange or more area for the gases to go in and out of the leaf. We sometimes just call this the leaf space. You can kind of see how the gases and so forth could collect in there, the gases for photosynthesis and respiration that are going to be transported, and also transpiration, by the way, and those things are going to be transported in and out of the xylem and the phloem. Now there are some modifications to leaves. What I've drawn here is what we call mesomorphic leaves, meaning that mesomorphic is referring to water relations in the environment. So mesomorphic means moderate amounts of water. It also basically means moderate amount of nutrients as well. And so a mesomorphic leaf is kind of the middle leaf. Now there are two other features. One's called xeromorphic and one's called hydromorphic. Xeromorphic means in dry environments, so either upslope or very sandy soils, and hydromorphic means wetland kind of environment. So some of the modifications that you might see are multiple layers of epiderm. So I might have many, in a dry environment, many layers of epiderm with cuticle, and sometimes really thick cuticle and sometimes really thick cell walls. The other thing is something called stomatal crypts, which I have not drawn here, but what would happen is the stomata would be actually back in the leaf surface. So they'd be inside the leaf, and there'd be like a little hole or inundation there that is usually surrounded by trichomes or hairs that then protect or keep the water from evaporating out easily, keep wind off of the leaf surface, which increases evaporation, and basically just kind of takes the stomatal gas exchange a little bit more internal to the plant. The last thing is usually they have very small and thick leaves. This just reduces the amount of surface area, not necessarily surface area per volume, but because the top layer has less stomata or no stomata, very thick cuticle, only the bottom layer will have many hairs, less stomata, and they will have those stomatal crypts. So that's kind of a common thing for xeromorphic leaves. Hydromorphic is the opposite, so they're in wetland environments, and they would have their stomata on the top. So what would happen is the stomata on the top would have to go through, the substomatal chamber would have to go through between palisade parenchyma. So what you would get is a line of a substomatal chamber that is going down between the cells, fairly commonly along the top, into the spongy parenchyma, which would then be completely enclosed. In other words, there would be no stomata down here. And that spongy parenchyma, to kind of keep it spread out, will have sclerids, or big fibers that are in there that are sort of keeping this from collapsing if something were to get on the leaf or something like that. And oftentimes it'll have trichomes on the bottom to protect from herbivory from fish. So those are just some common features that you see. Many times the leaf is very large and spread out on the surface of the water, has reduced xylem because it's in the water, so it doesn't really need to transport. Reduced support of stem, actually, also many times, because of course a lot of these plants are floating, which is why the spongy parenchyma area needs to be maintained to keep a lot of air or gases within there to help with the float. All right, so let's just look at the model here. This is a model of the leaf. Let me turn it this way. So this is just showing you, of course, well, you should be able to see that this is a eudicot leaf. That is, here's the palisade parenchyma here. This is showing you the stomata in the surface, and these are the veins. In this case, the colors are different than how I drew it. So what would be on the top? The xylem, and the phloem would be on the bottom. These would be the bundle sheath cells, and this would be the spongy parenchyma. The space that's right here, sort of in this space, right behind the stomata, is the substomatal chamber. Notice in this leaf diagram, they have stomata on the top and the bottom, so that is also possible. All right, so this is just showing you epidermal cells with the cuticle. Notice how the epiderm aren't usually green. They are not usually the photosynthetic part of the plant. The mesophyll is the big photosynthetic surface.